Regenerative Medicine and Longevity.
You must understand the kind of cells that are involved. First, the embryo. Fertilization of an egg will create an embryo. That is the beginning of life. As the embryo develops, it develops into a blastocyst. After the first few days, the blastocyst has cells on the inner lining, on the inside of it, that are basically called embryonic stem cells. These embryonic stem cells at that point of life can develop and differentiate into any type of cell, tissue, or organ. But the cells that we are interested in, the stem cells that we are focusing on, are essentially the stem cells of the mesenchymal stem cells. These mesenchymal stem cells that differentiate into tissues that are basically bone, cartilage, and muscle are quite important in our evaluation. However, it's important for you to understand that these basic cells that I have just mentioned and the mesenchymal stem cell differentiate only into its line of tissues and organs. But remember, there is another type of cell, an induced pluripotential stem cell, or induced pluripotent stem cell. This is an embryonic stem cell that we can create or we can develop through certain techniques. One technique is that of transfection of a mesenchymal stem cell with certain genes, such as the OCT4 gene, the SOX2 gene, and the E-MYC gene, which is spelled, or the C-MYC, C-M-Y-C, and the KLF4 gene. By putting them together and having them enter this cell, the mesenchymal stem cell, those four genes will convert the mesenchymal stem cell back into its embryonic state, the ideal cell for regenerative medicine. Because an embryonic cell, an embryonic stem cell, can differentiate into any kind of tissue that's needed in the body. However, you must understand that there are downsides currently to this process. The first downside is that the CMYC gene is a form of an oncogene, and this gene can develop cancer, unfortunately. The second downside is the transfection of a cell, or a mesenchymal stem cell, with these genes, utilizing the science of a retrovirus is doable, and it has been done. However, the retrovirus, just as if a virus would infect any type of cell, it would infect the mesenchymal stem cell with the genes, since we put the genes into the retrovirus that we want to transfect into the mesenchymal stem cell. Must be understood that the retrovirus has some issues in expressing certain oncogenes or even developing the formation of potential cancer cells. There are two new developments that are breaking this process wide open and eliminating the cancer possibility. The first process that has been discovered is that there is a ENT3A factor that is widespread and exists that can be placed into the 4 genes that can eliminate, not placed into, that can eliminate the CMYC gene in this group of genes that will convert the mesenchymal stem cell into an embryonic stem cell, which is the ideal cell for regenerative medicine. Does not form cancer. It does not form cancer. These 4 genes now would be the OCT4 gene, the SOX2 gene, the gene that I just described as the ENT3A gene, plus the KLF4 gene, another gene, maybe the NANOG gene, would be added to that, that would push the mesenchymal stem cell into the mesenchymal state. Great news, no cancer developed from that. We're on our way, found an answer for that problem. The second answer that we have found is the problem that existed, as I've mentioned, with the retrovirus in the formation of cancer. There is a new technique called electroporation, which absolutely does away with the need for the retrovirus. What it does is it invokes a electrical impulse into the cell, into a solution with the mesenchymal stem cells there, that will actually, it would be a millisecond of electrical impulses that will cause micropores to exist in the mesenchymal stem cell membrane, the phospholipid membrane. Now, this pore is basically an electrical current, also would be involved with that, that will enable DNA, which is charged particles, to enter the cell through that process. And since DNA are these genes that we've been talking about, voila, we have found our mechanism of eliminating the retrovirus in the transfection of mesenchymal stem cells. A wonderful breakthrough. A breakthrough that will be very meaningful for mankind and be the basis for successful regenerative medicine that will not cause cancer. So, what do we have now? Well, we now have stem cells that have been turned into an embryonic cell, an embryonic stem cell, an induced pluripotent stem cell, which is basically an embryonic stem cell, can turn into any tissue. What do we have now? We have extracellular matrix. In the development, in the extracellular matrix, there are growth factors. The growth factors are basically cytokines or signaling mechanisms that will direct the cell, the embryonic cell, to grow and differentiate into the kind of cell that we need. And this is done by a signaling mechanism, which we're not going to go into at this time. The other important breakthrough is the scaffold in which the stem cell, the matrix, are involved with. This scaffold could be basically formed into any kind of organ, any kind of tissue that the stem cells can grow on or within. And remember, these scaffolds are like sutures that dissolve. Scaffolds can be in the form of polysaccharide scaffold, protein scaffolds such as collagen, fibrin, even silk, and also a number of other scaffold materials that have been developed in the form of polymers, and there have been major breakthroughs in those areas. So, what do we have? We have the platform now with all of the factors that we need to make regenerative medicine work. This is the new field. Understand that. There are major centers throughout the entire world that are leaders in each one of these areas. We will identify these centers and post them in the near future. So stay tuned. This is viable. So we're on our way. We're on our way because you must understand that tissues are damaged. Man's tissues, man's cells, by aging, by injury, and by disease, etc., etc. These stem cells that I'm talking about now can repair, rebuild, and regenerate new organs. What an exciting time in our lives. What an exciting time now. And we are on the forefront of this wonderful, wonderful idea.